Welcome to the wonderful world of biology. This video will cover the basics of plasmids, such as definition, properties, classification, and finally, functions of plasmids. Before knowing about plasmid, one should know the definition of the chromosome. Chromosomes are the genetic material that encodes for essential information that specifies the organism. Non-growing bacteria usually contain a single chromosome. When growing bacterial cells could contain multiple copies of the chromosome depending on their synchronization of chromosome replication with cell division. After knowing what is chromosome, now we can define the plasmid. Plasmids are independently replicating genetic material that does not encode for any essential genetic information. Sometimes plasmid does not encode for any obvious characters apart from their replicative nature. Such plasmids are called cryptic plasmids. Other plasmids do encode for certain selective characters such as antibiotic resistance. More information on different types of characters encoded by plasmid is given into the functions of the plasmid segment of this video. Plasmids are commonly present in bacteria and also found in lower eukaryotes such as 2-micron plasmid of yeast. Interestingly, the plasmid can exist in two states. 1. Physically separated from chromosome called plasmid state, and 2. Inserted into the chromosome by recombination called chromosomal state. Next segment is categorization of plasmids. In earlier times, plasmids were simply classified based on their genetic marker. A genetic marker-based classification system has divided plasmids into classes such as self-transmissible plasmids, resistance plasmids, hallucinogenic plasmids, etc. But, as newer plasmids were discovered, this scheme becomes crowded and confusing. In 1976, Novak has proposed a systematic classification of plasmids. According to this system, plasmid name should be starting with lowercase p, followed by two uppercase letters as the identifier for the discovery or the discovering laboratory, then a number for particular plasmid. For example, PUC 18. The Plasmid Reference Center in the United States allocate and maintain identifiers. Next segment is properties of plasmids. Bacterial plasmids are usually double-stranded covalent closed DNA. Single-stranded circular DNA plasmids, linear plasmids, and RNA plasmids also exist in nature. The size of the plasmids ranges from 1 kb to greater than 400 kb. Plasmid copy number is the total number of plasmids a cell can sustain. For example, large plasmids are under stringent replication control and have low copy numbers per cell, while small plasmids are under relaxed replication control and have high copy number per cell. The copy number of plasmid reflect the replication control. Replication control is exerted by RNAi, interons, ctRNA. Bidirectional replication and rolling circle replication are common modes of plasmid replication. Plasmid having similar replication mechanism does compete with each other to replicate, hence they both cannot sustain in a single cell. Such plasmids are incompatible with each other. Another property of plasmid is the host range. If a plasmid can exist in a limited closely related organism, it is known as a narrow range of plasmids. If a plasmid is found commonly in diverse cells, it is called broad-range plasmid. Last segment is functions of plasmid. Depending on the size of the plasmid, it can encode single or multiple proteins. Certain genes present on the plasmid can provide a selective advantage to organisms sheltering that plasmid. For example, it can provide resistance to bacteria against heavy metal and antibiotics. It can provide enzymes to utilize unusual carbon sources. It can allow bacteria to synthesize antibiotics, toxins, or proteins that increase virulence. It can also code for hormones that could form tumors in the host. As the end of the video approaches, I would like to thank you for your attention.